I am going to present to you my abstract, which title Special Variability Attributes of a Ultisols hydrosequence with aid of portable X-ray fluorescence spectrometry. I'm Fernanda² Bócoli, and here are my co-authors. I am going to do a little introduction to you. The proximal sensors are becoming very helpful for soil-related studies because of being rapid, with low cost of analysis and no generation of pollutants. The spatial variability of attributes in a profile, like elemental contents, explored in few studies. Here I cite some of them, but the spatial dynamic of soil profile attributes can aid in numerous studies, like in pedology, for studies of genesis and elemental or mineralogical dynamics, or agricultural management, because when you manage a soil, you have to know the soil, and this type of study can permit you to know better your soil in a more detailed scale. Hartman and Ms. Nunning proposed in this regard the soil digital morphometrics that analyzes the differences along the soil profile with proximal sensors aid. Here are some examples, like the portable X-ray fluorescence, the magneticity limiter. This study evaluated the spatial variability of 3 Ultisols piles in a hydrosequence via portable X-ray fluorescence spectrometry. The hypothesis of this study is that the profile position on the landscape rules the attributes variation. Then the objectives is to investigate causes of soil profile differences along the hydrosequence and characterize and evaluate attributes more variable in the Ultisols with the use of pXRF and magnetic susceptibilimeter plus the morphological description. With this, we want to understand the soil profile genesis and elemental and magnetic minerals interactions. Here is the study area located in Brazil, Minas Gerais State, Labras Municipality. Here is the location of the soil profiles and its elevation and its position on the landscape we can see on this 3D map. The soils that were analyzed is a thick hydrodote, a thick haplodote, and a incept haplodote. Here is how the samples were collected on a grid of 20 to 20 centimeters. The analysis with the pXRF take about 10 grams of arterial thinner sieved to 2 millimeters. The spectrometer was configured on trace mode and read the samples in triplicate, taking about 60 seconds. The spatial analysis using the data generated by the pXRF, that is the total elemental contents of the samples, used the technique of interpolation with the aid of a multi-level baseline of the software kits. Then 3 Ultisols profiles were analyzed in native Atlantic forest, which parent material is the gneiss. Here are the results from magnetic susceptibility. First of all, we can see that the highest values are on the surface horizons. For the typic haplodote and the incept haplodote, we can see that they receive the materials from other areas. In this area, we have many soils derived from mafic rocks that contains on its compositions magnetic materials as magnemite and magnetite that can increase its magnetic susceptibility, mainly for the incept haplodote that is in the lower third here, receiving more material. For the typical hydrodote, we can see that its highest value is on AB horizon, but is on the surface too. This can be because of its upper position on the landscape. say that favored its better drainage and the formation of magnetic materials more than the other profiles that don't have a drainage like this profile. This can cause a lithological discontinuity from this profile to the others. Here we can see that the insect haploidot has highest total elemental contents than the typical haploidot, except for the potassium, that this typical haploidot has highest contents. And defined patterns we can see only for the insect haploidot. On the other profiles we can see the contents very mixed, don't have clear patterns, but only for these elements. When we see the zirconium and titanium contents, they are used by other authors like Mancini and his co-authors in 2021, used these elements as tracers of parent material, what can mean that its parent material from the surface is different from the subsurface. Here, another possibility for the magnetic susceptibility being higher in the surface is about the iron contents that are higher in the upper horizons than the lower ones. Another evidence of a lithological discontinuity from the typical haploidot for the other profiles is its contents, mainly of aluminum and silicium, much higher than the other profiles. And when we see the zirconium and calcium is much lower than the other profiles, what is another evidence from these differences. And the elemental contents depending on the dominant soil forming process. Here in the case is the relief and the slight variation on soil parent material. Mancini and his co-authors found a large variation on the composition of the gneiss, an area next to this one here analyzed. Then the conclusions of this study is that the profile differed by their color and elemental distribution. The soils have the same parent material, but the area seems to have a lithological discontinuity from the typical haploidot to the other profiles, and the insect haploidot may have a deposition of sediments from upper areas of the landscape. The spatial analysis shown to us that even morphologically homogeneous horizons of the same profile, there might be a wide variation in the distribution of elements as iron, silicon, and aluminum. I acknowledge funding agencies, FAPEMIG, CAPES, and the CNPq, my institution that I come from, the Universidade Federal de Lavras, and the post-graduation in soil science, and this event organization. Here are my references, and thank you. Any doubts? Here I use my contact if you have some. Thank you.